Good afternoon, and thank you for joining us for the 2020 Avondale House Unluncheon, presented by Texas Children's Hospital. My name is Michelle Wisely, and I'm the Development Director for Avondale House. Due to COVID-19, we, like so many other Houston nonprofits, have had to reimagine our biggest fundraiser of the year, and we are so thankful that you all are joining us in this virtual space. Since we cannot be together today, we ask that you take pictures, selfies of yourselves enjoying our virtual event and send them to us at info@avondalehouse.org. That email address again is info@avondalehouse.org. We also encourage you to share them on social media and tag us. Also, don't forget to please enter any questions you have for Dr. Zogby in the comments section. This event would not be possible without the support of our amazing sponsors. As I said, Texas Children's Hospital is our presenting sponsor this year. Our platinum sponsor is Westwood Wealth Management, and Hogan Lovells with Ann and Greg Hill. And Mary Weldon and Guy Buckley are our gold sponsors. Our silver and bronze sponsors are HEB, Fred Steves, Carol Keeney Harrington, Ann Turnbaugh and Steve Ulrich, Crystal and Michael Williams, Christina and Steve Vetrano, Cheryl Verlander and Chuck Brock, Ashley and Christian Nelly, Community Health Choice, and our wonderful event co-chairs, Kathleen Mahome and Milan Jomrich. We truly could not have done this without all of you. At Avondale House, we proudly serve more than 100 individuals who are severely impacted by autism with the resources, education, and training to meet their fullest potential. We do this through a day school, an adult day program, and a residential program, and we also have a new supported employment program. Today, 1 in 59 people are affected by autism, so the need is great, and Avondale House is the only organization in Greater Houston serving the most profoundly affected individuals with autism from early childhood through their adulthood. Our vision is to expand our capacity to answer the call to even more families in need. Your contributions to our event today will support this vision. Please go to the donation link in the comments to find out more about that. We are privileged today to have Dr. Huda Zogby with us. Dr. Zogby is a professor of pediatrics, molecular and human genetics, neurology and neuroscience at Baylor College of Medicine, an investigator with the Howard Hughes Medical Institute, and the founding director of the Jan and Dan Duncan Neurological Research Institute, where we are filming today. Dr. Zogby is a multiple award-winning leader in the medical community, and her group's research is on the forefront of gene research in relation to autism. Now don't forget, as Dr. Zogby is presenting her talk today, to submit your questions in the comments for us. And with that, I will hand it over to Dr. Zogby.
Thank you very much, Michelle, and thank you everybody for tuning in today to hear this replacement, if you will, for the luncheon. I have to thank Kathy Mahone and Milan Yamrich for inviting me to be a member at today's luncheon. I used to know Alex, and I really know how much he enjoyed the Avondale House, so I thank you so much for allowing me the chance to visit with you today. So what I thought I'll do in the next 10 minutes is tell you a little bit about how far we've come to try to understand autism. Where are the clues coming from? What are we doing today, and where we hope to go? And of course, that's a lot to cover in 10 minutes. So I will just hit the highlights, and I will trust that you will ask us a question for me to elaborate on anything that may not be very clear to you. As some of you might know, I'm a child neurologist. So I actually started my career in the clinic. And back in the days when I was trained in the mid-'80s, autism was really rare. We did not encounter many people with autism back then. But I did see some people with what we call syndromic autism. And what I mean by that are people who have different neurologic problems, but one of their symptoms is autism. So they have autism as one of a multitude of other problems, cognitive problems, motor problems, seizure perhaps, and so on. And that really captured my attention. I was pretty much intrigued by a disorder called Rett syndrome, where just like in autism, where the children appear healthy for the first year or year and a half, and then they regress, and then they have problems, they lose any language, or they never really communicate, and they have repetitive movements, and they have they become quiet and withdrawn. I was drawn into that disorder, and I decided to study it. And what I'm going to share with you today is how the study of this syndromic disorder is teaching us something about what I call the more common form of autism. The more common form of autism is not syndromic. It's really the classical feature of autism: lack of social interaction, problem with communication, and some repetitive movements, and sometimes with or without difficulty learning. So studying a disorder like Rett syndrome, which is like most of autism, is sporadic, taught us something very important about other forms of autism. The first thing it taught us is that a disease can be genetic even if it's not inherited. And what I mean by that, I mean that if an individual is affected, but they didn't inherit a gene mutation from either their mom or dad, it actually happens sporadically in them. And there were some other disorders that became shortly after that known to also be genetic and sporadic. And I would say that era in the late 90s, early 2000s really opened up the discovery for a variety of forms of syndromic autism, where basically you now know that there are gene mutations that underlie the causes of these very complicated disorders that have autism as one of their features, but they also have many other neurologic problems. Why is that important, and why am I sharing this information with you? The reason it's important, it hinted that a lot of forms of autism may actually be due to a gene mutation. And it told us that really autism is caused by biologic or genetic factors, rather than us worrying, is it how someone raised this child, or is it what someone did during a pregnancy, and so on and so forth. Majority of it is determined by that. The second reason, which is really important, as we studied these genes, that each one of them causes severe syndromic neurologic problem with features of autism, we quickly learned that there are some minor mutations, some mutations in the same genes that cause Rett syndrome, or cause Phelan-McDermid syndrome, or cause other syndromic forms of autism. The same genes, if they have a milder mutation, which means a mutation that doesn't really inactivate the function of this gene completely, just partially disable it, if you will. What we learned from that those same genes can now form what I call the classic autism. That is a milder form than the syndrome, showing only the features of autism. And that's where our research in the last 2 to 3 years has led us, in that we began to look at these more milder mutations and finding that those milder mutations may cause the isolated or non-syndromic autism. Why is this important? It's really important because it tells us that the mildest form of disrupting neuronal function is going to manifest, if you will, in autism and mild intellectual disability or behavioral problems. Only when that impairment is more severe, you start seeing these other complicated motor impairments, as well as seizures and other things that these syndromes are characterized by. And the other thing it tells us, because we know that the majority of people with autism, we really don't know what genes causes them. What we look when we find mutations in these syndromes in some people with autism, that's only the tip of the iceberg. We still have to discover all these variations in the DNA that can cause the milder forms, or the isolated, or the classic form of autism. But this is important because now that we know that social behavior, communication is really at the top of our biological functions and thus most vulnerable. The first thing to give you a symptom, if you have even the mildest of genetic change, now that, we know going forward where to look and how to look. And I imagine as we study these variants more deeply and as more and more sequencing is done, we can discover such mutations now, as long as we keep in mind they're going to be very mild. The other thing is we know now how to study the effect of these mutations. 10, 15 years ago, it was really extremely hard to study the effect of a small change in the DNA, except if you have to do it in a mouse, and it's quite costly. But now there are other model organisms that at least you can determine if this genetic variant is causing a consequence in a lower model organism, whether it's a zebrafish or a fruit fly. Then you can go back to the mouse, and this way you can screen a lot more variants. So I imagine the next few years are going to bring us a lot more candidates that may explain to us some of the determinant of autism, if you will. The other thing we learned is that many of the genes that cause autism, whether is it syndromic or not, they're important for the function of neuronal communication, what we call synaptic function, where two neurons meet and communicate. So we're learning more and more that no matter what the gene does, the end effect is affecting that synaptic function. And we learned also that sometimes you do not need to mutate a gene. You might just have a missing copy or an extra copy. That means it's just either a deletion of one gene or a duplication of a gene that might cause that. So you have either too little of the protein made by that gene and too much of the protein made by that gene. And again, if it's a 50% reduction when you use a copy or a 50% increase when you duplicate a gene, that will give you some of the more severe forms. We believe perhaps there are changes that may only affect by 20% the level of a gene, reduce it from 100% to 20%, and that's going to be enough to give us, again, the classic or non-syndromic form of autism. This is really hopeful. Why is it hopeful? Because if something is only decreased by 20%, we can imagine easier ways to bring it back to normal and hopefully bring therapies to those people affected by that. So on the topic of therapies, where are the therapies? How they're going to come from? What have we learned? There are really two classes or three classes of therapies, I would say. Sometimes, if we're lucky and we figure out what a gene does, and we can find a small molecule, a drug, that may affect the activity of that gene, that may be a good way to go. I'll give you an example. Some forms of syndromic autism, like tuberous sclerosis, we know that a particular pathway, the mTOR pathway, is overactive, and we know that a drug that inhibits that pathway can actually help the epilepsy in these individuals. And there are studies ongoing now to see if the same drugs will affect social behavior and cognition. So that's one class of therapy. Those are the lucky ones, where you really find it off the shelf, it's already FDA approved, and you can test it. But this is only going to work for those genes for which that pathway is affected. The other approaches, it depends on the severity of the disease. Sometimes gene replacement therapy, as in gene therapy, is necessary, as we've learned, for Rett syndrome, for example, where you have to actually replace the protein. Nothing else will make up for it so far. A third class of therapeutics, when you have too much of something, as in the case of the duplication of the Rett syndrome gene. Doubling the Rett syndrome gene creates another disorders that's characterized by intellectual disabilities and autism. And having too much of that causes that disorder that's progressive. There's something called antisense oligonucleotides, or ASO, which is a small piece of the DNA that you can give it to the individual, and that can lower the copies of the RNA made by the duplicated gene and will normalize it back and make them better. Now, we've shown that in mice, and we have to be preparing for eventually translating that to humans. We're not there yet, using that for a treatment in human. But you can imagine if we're successful and we can do it safely, where we lower the protein back to normal level, not below normal level, we can then apply the same approach to many forms of autism that are caused by gene duplication or an extra copy of a gene. So these are all examples of therapies, and we know ASOs have been tried in other disorders like spinal muscular atrophy, where they've been successful and safe. So we know we can give them to children safely, but for each particular disease, we have to give them where the amount is just right. So these are all different types of therapies that are emerging, and it's really exciting. I was talking to the director of the National Institute of Neurological Disorders, and the NINDS is going to be all about moving these therapeutics, whether it's gene therapy or ASO-based therapy, any form of therapeutic, because so many discoveries have happened in the realm of autism and syndromic forms or non-syndromic that we are now ready to move that in the next few years. Besides all of these, there are other forms of therapies, because as I mentioned to you, many of these mutations affect how neurons communicate. It turns out you can bypass a deficiency of a gene by going straight to activating a network within the brain, a small area you can activate, and that area might affect how the individual will learn or how the individual will move. And this is new and emerging. Again, research in animal models is promising that activating particular parts of the brain is helping the models of these diseases learn better. And we know that deep brain stimulation that has been used in this area has been used in humans for other applications, for sure Parkinson's disease, but also in children for some forms of epilepsies. So we believe that this is another emerging area of therapeutics. And you can do that specifically for a particular part of the brain and improve the function of that part of the brain. We don't know yet if there is an area we can stimulate to improve communication and social behavior, but these are emerging studies, and the Brain Initiative is going to help us identify those areas so that eventually they could be used therapeutically. The beauty of network modulation rather than gene replacement or other forms of gene-specific therapies is that this may apply to many forms of autism, irrespective of what's the underlying cause. But they're going to take a little bit longer to develop, but I think they hold a lot of promise. And one last area I want to bring out, which I've been really impressed to see how effective it is. I shared with you that we've tried the deep brain stimulation in the rat mouse models, and we found that the animals could learn better. It turns out if you do excessive training of the animals and behavioral training, you could almost achieve some of that good benefit you achieved with the more invasive approaches. And I know all of you are doing that because all of you have had experiences with specific behavioral therapies and physical therapies that made a difference for the individuals with autism in your lives. But what really impressed us from the studies we've been doing is that the earlier these treatments are started, the better the outcome is. So I would really urge that anytime we see individuals that may not have all the typical developmental milestones, and there is any concern about autism, is to really diagnose them as early as possible, because the earlier the intervention, we're seeing that the outcome can, at least in the models of Rett syndrome, can be much, much better. And I know all of you are doing that, but I want to mention that what you're doing is worthwhile. And you may not see the effect quickly, but you're reaping some benefits by improving some activity or delaying regression or preventing regression of some activity. And I know during this time of the COVID pandemic, this is extremely challenging. It's really, my heart goes out for any individual trying to handle someone with autism or intellectual disabilities or any neurological problems, because they're not going to understand why this lockdown, and it's going to be really challenging. And the physical therapy axis and behavioral therapy is not the same, but anything we do, I'm sure, will make a difference. And this is what I shared with some group of friends of the Institute, that yes, there is a pandemic, but those other neurological diseases, they're here, and they're going to stay with us when there's finally a vaccine for this pandemic. So this is why I'm excited that we're going to open up the Institute this week and get people back to work, to continue to work in this area. So with that, I will maybe stop to allow some time for questions.
Great. Well, Dr. Zogby, thank you so much for a glimpse into the cutting edge research that you and your team are doing. And we at Avondale House feel so privileged to have been able to still hear you speak in this virtual space and give hope to our community who are especially struggling during the COVID-19 pandemic. So thank you for those comments as well. We have lots of questions coming in from our viewers, so let's get right on over to those. The first question is, do you have insight into why males are more affected than females with autism?
That's an excellent question, and thank you for asking it. I have been really thinking long and hard about that. As many people know, when it comes to classic autism, there are about 5 times more people with autism who are males than there are females. And in fact, this week, I put together a proposal with colleagues who are also thinking about this problem, colleagues at University of Washington in Seattle, Johns Hopkins University, and UCSF. And the 4 of us are going to team up, capitalizing on all of our strengths to try to figure out why it is. I can tell you some thoughts why it might be. One possibility that there may be some changes on the X chromosome, which typically, because if a female has 2 X chromosomes, therefore, if there is a change on one of the X chromosome, it's going to be only in 50% of that female cells, and therefore you may not see an effect. But in a male who has a single X chromosome, you're going to see more effects of that. With all the sequencing efforts, the X chromosome has been elusive. It's been really hard to capture using the existing technology, the genes carefully, and annotate them from the X chromosome. So one of our colleagues at the University of Washington is going to be focusing on that, looking at 50,000 samples and try to test that hypothesis. Other potential differences could be because of hormonal differences. In our brain, there are genes that respond to estrogens, and there's genes that respond to androgen. What we don't know is in the parts of the brain that's critically important for social behavior and communication, how are there such differences? Are there genes that are uniquely androgen responsive or estrogen responsive, and how do they differ in the male and female brain in a healthy individual or in an individual with autism? So all of us in this group, we're putting together an application to study that. All of us are going to look at these various angles, and then once we identify the candidate factors responsible for that sexual difference, here at the Institute, what we hope to do is test that in animal models and see if we can begin to show male only versus female in animal models to prove that.
Wonderful. We have another question. Do we know if epigenetic alterations contribute to autism?
That's another interesting question, just for everyone to know what's an epigenetic alteration. So genetic alteration is either when you delete a gene, duplicate a gene, or change the DNA sequence in that gene. And that, of course, will lead to altered protein function and will change the activity of, or lose the activity of the protein. An epigenetic alteration is not a change in the sequence at all. The sequence is the same, but it is a change in how one particular base pair of the DNA, cytosine, that base of DNA can be modified by adding a methyl group to it. And that modification is important. Although it doesn't change the sequence, this epigenetic modification is important because it can change the expression and the level of a gene or a protein. And I mentioned to you that we find that small changes, as much as 20%, are consequential. So yes, epigenetic changes could contribute to autism. They're harder to find because you have to look at the right cells in the right brain region, because epigenetic changes, how these cytosines are modified, they will vary whether it's in a blood cell or whether it's in a brain. Even within a brain, it will vary depending on the cell type. So you have to really be looking at cells that we know are important to features of autism and look for epigenetics changes right there in people affected with autism to find them.
Okay, wonderful. We have a two-part question that came in from Scott. The first part of the question is, can you comment on the impact of the microbiome on the manifestation of ASD?
This is the best set of questions I get. Okay, that tells me I'm dealing with a highly sophisticated audience, and I appreciate you. So the microbiome, as many of you know, is the host of bacteria that is inside us and on us, right? There are bacteria on the skin, and they are quite different, whether you're looking at the upper surface of your arm or the lower surface of your arm. There are bacteria in the mouth, there are bacteria in the GI tract, they're everywhere, and they're really important. There are more bacteria on us than probably there are cells in us. Why are they important? Because for certain functions, they actually provide chemicals, if you will, that you may not get from the food. They may metabolize things, they may provide things that our cells use and allow us to respond. And we are learning something really important. There are many cells in our, let's take the microbiome in the gut. We know there are many cells in our gut that actually secrete neuropeptides or neurotransmitters or hormones that have receptors in the brain. So there's talk from the gut to the brain, and we know the microbiome helps so many gut cells in so many ways, and I wouldn't be surprised if there is some contribution. Of course, the contribution is probably a combination. There's probably a genetic variation in an individual combined with a microbiome interaction that might lead to certain symptoms. And therefore, altering the microbiome might be helpful, but it would not be sufficient. But it is a path for us to investigate. What's more important is to really investigate how the microbiome can affect an individual response and features of autism.
Okay, great. And then his second part of the question is, can altering the microbiome potentially benefit those on the spectrum?
So again, as I mentioned, this probably is not going to apply to everyone. There are interactions. I think a healthy microbiome is typically obtained from a diverse. The most important thing about the microbiome is to have a robust microbiome. How you have a robust microbiome is, of course, by eating the diversity of foods, including healthy fruits and vegetables, high fiber content food, and so on. So having a healthy microbiome is really important. Whether a particular therapeutic additional microbiome, if a person is unable to eat or ingest, would help, this has a potential, but this hasn't been studied extensively.
Great, thank you for that. Why do some people with autism never speak?
Part of the reason, so I think of autism really as a true spectrum, from an individual who can speak, but they're maybe more reserved, they prefer to be isolated, they prefer minimal social interactions, all the way to those, of course, who might speak a little bit, then lose that, to those who never speak. As you move across the spectrum, what we think is happening is that synaptic function that's important for the area for speech and language acquisition and communication may be compromised. And why might they be compromised is there's probably some genetic underlying variations that have led to that. So I do believe the more severe forms, just like we talk about syndromic autism, the more severe forms are probably those who never got speech and never acquire speech, are probably because they probably have changes that are affecting cortical areas, synaptic function, more so than those with the milder forms.
Okay, great. Do we have any more questions? Okay, I think we are good. Dr. Zogby, thank you so much again for your time today to participate in our event. As you saw with the great questions that came in, you clearly engaged our audience with your talk, and we're so very grateful for the important research that you're doing to help solve the puzzle that exists with the neurological disorders, especially autism. And as we wrap up our virtual event today, I just want to thank everyone in our audience again. We appreciate your time and commitment to the mission of Avondale House. And I want to emphasize that when you donate to our agency, you're playing an integral role in providing students and clients a community in which they are welcomed and accepted for exactly who they are. You are helping to provide the resources, education, and training to foster the unique needs and talents of individuals with autism. Your support truly keeps our students thriving. Thank you for making a difference in the Avondale House community.
Thank you.
Thank you again.
Thank you.